Hi, this is Frank from Intel Minds, and today we're going to be talking about 1.2 Ultrastructures of Cells from the IBTP program for biology. This section will mainly focus on structures of eukaryotes and prokaryotes. Firstly, we're going to be talking about prokaryotes. From the name, we can see that pro means before, and karyon means nucleus, and therefore prokaryotes lack a nucleus. This is easy to remember as it's derived from Greek. So prokaryotes are single-celled organisms. They vary from 0.1 micrometers to 5.0 micrometers in diameter, and they include the kingdoms Eubacteria, which are classified as more traditional bacteria, and we also have Archaeobacteria, which are organisms that are found in extremely high temperatures, salt concentrations, or pH. And prokaryotes exist in different shapes, such as coccus, bacillus, spirillum, coccobacillus, and spirochete, which are more complex shapes, but you don't need to know that for this section right now. So here we have figure 1.1, which is the structure of a normal prokaryotic cell. We can see that there's a cell wall, which protects the cell and gives it its shape. We have the plasma membrane, which is found all throughout the cell body, and it is what controls the movements of substances in and out of the cell. We have the cytoplasm, which fills the whole cell, and it is the site of all the metabolic reactions, which include enzyme-substrate reactions. We have pili, which are not shown on the image here, but they would usually be drawn as like little spikes, and they are protein filaments on the cell wall that help in cell adhesion and also transfer DNA between two cells when we have two different bacteria cells interacting with each other. We also have the bacteria flagellum, which creates whip-like movements, and this creates the locomotion of the cell. We have 70S ribosomes here, which are the site of protein synthesis for prokaryotic cells. We have the nucleoid region in the middle, which is basically the circular DNA. It's known as naked DNA as it is not membrane bound, and it controls all the activities of the cell and also reproduction. We also have these small independent regions, which are called plasmids. They're small circles of DNA that include adaptive genes in order for the cell to exist over time and adapt to its situation and surroundings. And this allows it to it allows it to reproduce and be competitive in an environment over time. Next, we're going to be talking about eukaryotes. Again, from the name, we know that eu means true and karyon means nucleus, and therefore there is a nucleus for eukaryotes. Eukaryotes vary from 10 micrometers to 100 micrometers in diameter, and they include four kingdoms. The first kingdom is Protista, which are unicellular or multicellular organisms that have no specialized tissue. We have fungi, which have a cell wall made of chitin, and they obtain nutrition via heterotrophic absorption. We also have plantae, which are plant cells, and they have a cell wall made of cellulose, and they obtain nutrition as autotrophs, and this is due to photosynthesis. And then we also have animalia, which are animal cells, and they have no cell wall, and they obtain nutrition via heterotrophic ingestion. We also have the endosymbiotic theory, which allows eukaryotes to have much more complex structures than prokaryotes, and explains that eukaryotic organelles, such as mitochondria and plastids, first evolved from prokaryotic cells many years ago. So first we have figure 1.2, which is the structure of a animal cell. The animal cell has, different structures from a plant cell, but the main components will be talked about right now. So first we have the plasma membrane, which controls the movement of substances entering and leaving the cell, and it's approximately 10 nanometers thick. We have the cytoplasm, which contains enzymes and also holds organelles together. We have the mitochondria, which is a double membrane structure shown right here, and it's the site of aerobic respiration. We have 80S ribosomes, which have protein synthesis for the cell. We have the nucleus shown in the middle here in pink, and it's the largest organelle in the cell that contains DNA. It regulates cell activity and also controls reproduction in unicellular organisms. We have the nucleolus, which is a part of the nucleus, and this is where ribosomes are synthesized. Next, we have the smooth endoplasmic reticulum, which is shown here. We can see that it doesn't have ribosomes like the rough endoplasmic reticulum, and it's involved in producing and storing lipids and steroids. It is also the storage of calcium ions, which are not important for now. We have the rough endoplasmic reticulum, which can be easily identified as it is both further from the nucleus and also contains ribosomes on it attached to it. It transports proteins produced by the ribosomes to the Golgi apparatus. And then we have the Golgi apparatus, which is shown right here, Golgi bodies right here in green, and we can see that it packages proteins and lipids into vesicles, and it's a site of the synthesis of biochemicals such as enzymes. Next, we have animal cell only organelles, which are lysosomes. Lysosomes contain hydrolytic enzymes that destroy bacteria and also old cellular organelles. And we also have centrioles, which are located here in blue, and these help cell division by establishing microtubules, and these contain of 9 microtubules. Next, we have vacuole for animal cells. We can see it's much smaller, and it contains the needs for osmoregulation and isolation of waste. And next, we will move on to the plant cell, which has 2 different structures, which include the cell wall, which is much more rigid, and it's made of cellulose, and this protects the cell. We also have the chloroplast, which is similar to the mitochondrial animal cell, as it is double membrane bounded, and it's an organelle that contains pigments that are responsible for photosynthesis. Next, we're going to be talking about the main differences. So we can see that DNA is free-flowing in the cytoplasm for prokaryotes, whereas eukaryotic cells have membrane-enclosed nuclei. Eukaryotic cells have a much more complex system of membrane-bound organelles that divide the regions into cell. This is known as compartmentalization, which we talked about later. We have prokaryotic cells that do not have membrane-bound organelles, and we know this because membrane-bound organelles only exist in eukaryotic cells as they are multicellular. So compartmentalization involves membranes being used to separate organelles into different compartments, hence the name. This creates a greater efficiency in the cell as there's a greater flexibility on the number and also the position of each organelle, and it allows the cell to isolate toxic substances via the cytoplasm, and it allows different parts to be differentiated and specialized in different functions. And finally, we have electron microscopy. On figure 1.3 here, we can see that there's an electron microscope from the University of Waterloo here in Ontario. Electron microscopes basically use a beam of electrons to create an image of a specimen that's usually non-living. The image that is resulting is in a monochrome, which is black and white and lacks color like light microscope images. Finally, we know that the resolving power is around 0.1 nanometers, which is really high, and it magnifies objects about 500,000 times or more, and it results in these very detailed images in contrast to light microscopes. And there we have it. That's section 1.2 Ultrastructure of Cells from the IBDP program. Thank you guys for watching, and be sure to subscribe and like the videos in order to hear more from Intel Minds.